Wow. That's a, I think Zoom meeting just give us the sign to start. Good morning, everyone. I'm very happy to see so many faces here today, and I'm happy that we didn't accept more participants because if not, the room would be completely, people would be sitting on the stairs. Thank you very much. My name is Carlos Sanchez. I work here as a postdoctoral researcher at the university in the group of applied chemical ecology with Professor Sanchez. If you need anything, please come to me, or if you have any other questions, you can reach Joshua, that is our right hand and the guy who has been helping us with all the logistics here. Also, if you have any help or any question, you can reach Johanna. Where is Johanna? Johanna is back there. And also, Toby is also here. So those are guys, local guys, that they know everything, bathrooms, where to have lunch, for example, or what is going to happen every day. Okay. But if you have, also you can reach me if I'm somewhere around. Just ask me please. Good. Thank you very much for being here. And I would like just to start by saying this is a dream that we have with Nicolas. Professor Nicolas is here from Colombia, from the University in Afid. We end up trying to talk, like, I don't know, 6 years ago, probably, starting to cooperate with Colombia, between Germany and Colombia, and thinking what to do in the future regarding chemical ecology and how to bring chemical ecology also to Colombians. As I was here doing chemical ecology, so we got in contact, and then we decided to start trying to get some projects done. And one of these projects is a cooperation between Colombia and Germany. And within this project, they allow us to do these workshops. We did a chemical ecology course in Colombia last year that Professor Boneller came with us, and also Jonathan Gershenson from the Max Planck Institute of Chemical Ecology, and Ricardo from the Neuchâtel University. And it was really nice and successful there. And the second step of this project is this workshop that we want to offer. And the main idea of this project for me was when 12 years ago, that I was facing the basics or the beginning of metabolomics. When metabolomics comes to me, I was really looking for a workshop like this, that people guide me from the beginning what to do with metabolomics, how to play with these tools, how to design my experiments. So all the comprehensive metabolomics pipeline. And this is that was our idea with Nicolas. This is what we got together all these magnificent and excellent researchers, some of them the developers of the tools. And we hope you enjoy the day this week. It's going to be an intense week. I know that. I know myself that. But I hope you enjoy it, and please feel free to have fun. This is going to be more interactive. This is not going to be like I'm talking all the time here, or they are talking all the time here. This is both. We will have a conversation. Okay, these are not talks. We will have a conversation, and the idea is that if you have a question, please feel free to raise your hand and to interrupt us and to ask whatever you want. Okay? The more we interact, I think it's better for us, because then we can solve and we can continue in the next steps of it. Okay, what we have today. So this is not working. This is our program today. So I will start today with opening and introducing a little bit what is chemical ecology, and this is part of the introduction what I'm doing right now. Then we will have Josh introducing us about experimental design and how to really do a good experimental design, because this is really missing in many projects. Then we will have a coffee break. Coffee break for all of you will be here in the foyer. You already saw some tables there. They will bring some coffee and some cakes. And then we will please enjoy it. And also we will have time to chat each other and to get to know each other. So please enjoy this half an hour to have a nice break. Then Josh will continue, and then we will have lunch. For lunch, people probably will ask me, "Oh, Carlos, where I can have lunch?" So as not everyone is allowed to go to the cafeteria of the university, you need a special permission for that. So we will not allow to make permission for all of you. So luckily for you, this area is a university area, and as you can see in the surroundings here in the Sulpicius Plaza, you have a ton of possibilities for food, veggie food, meat, hamburgers, big restaurants. So please feel free to choose and get groups and go there and enjoy. You have 1.5 hours, so enough time to grab and listen. Then in the afternoon, I will go again and I will introduce you a little bit how I think, and actually that was a good exercise, how I think we should do sampling and sample preparation. Of course, I'm probably missing so many things in that talk, but this is actually nice that you're here so you can help me to make it more rich, this sampling and sample preparation. Then we will have a coffee break, and then we will have in a remote connection, Allegra, Professor Allegra from the University of Denver. She will give us a nice talk about sampling, data acquisition, and about native metabolomics in Eco-Metabolomics. So we will join with her. She's in the States, and she will join us around 3, and then we will listen to what she has to tell us for today. Good. Any questions regarding the program for today? No? Great. As I told you, I would like to first thank Nicolas for believing in this crazy idea of gathering together and building a project in order to cooperate between Germany and Colombia. And so far, so good. So we are successful. We have only one more step, that is try to write a project to go for DFG or BNDF to get some money for the big project. And of course, thank you also to Professor von Eller for allowing me to do this within his group and within the university. He was the person who said, yes, Carlos, go for that. It's a crazy idea, but go for that. Let's try it, and it worked. So he is the director of the Aquatic Chemical Ecology group, the group that I work with. Also, thank you very much. This for some reason is not working. To Joshua for all your help and all your support in this to organize. You show up in the right moment, the right day in my office, and luckily for me, this is possible thanks to him as well. Thanks to the university for providing this space and for providing some of the resources for this workshop. And very important, the BNDF, who was the one who was paying for all of this, for paying for all the hotels, for the dinners, for all the trips, for everything. So you have to thank this BNDF. So send a letter to the BNDF for this. Good. And we also have to, of course, thank the Minister of Science from Colombia that helps Nico to come here to join us. And of course, thank you to all the speakers. And I will just, you know them. I think you checked their faces and who are they already. I will just go very fast later to each one of them because I really want to thank to all of you. It was a massive registration. We have more than 120 registered people. And at the end, we end up only accepting 65. So we were really very, very happy for all the overwhelming response from all of you. We have, this is just a little bit of a statistics, who are here sitting, who are you? We have all different levels, academic levels, from master, bachelor students to scientists, postdocs, professors, research scientists, technicians. We also have, I'm sorry, I couldn't put the technicians on the second page here. But you can see that the biggest is postdoctoral researchers and PhD students. So it seems metabolomics is still in a higher range. We want to put a little bit more down, bring it into the masters and the bachelors, because we think it's important that they also get to know it from the early stage. We have the knowledge of you guys. We have, most of you have basic knowledge. Very few have no knowledge, and of course, there's one who has extraordinary good, of course, knowledge. I want to know. I think I know who's that. And of course, as we want everything balanced, we are very lucky that it's very, very balanced. Of course, we have almost 50-50 male participants in this stuff, and we are very happy with that. Good. Let's start with my talk. Okay. So I want to start asking you, what do you think is eco-metabolomics? What do you think is eco-metabolomics? Have you heard before about eco-metabolomics? Don't be shy.
But it's the study between the environment and the molecules and the organisms in the environment. So it's the connection.
We have a long connection. Isn't it? We have all the ecology part and the metabolomics part. And then actually that's what Sardans, who was the person who established this term, eco-metabolomics, I didn't invent it, Sardans invented it. He said, aims to analyze the metabolome, the total number of metabolites, and its shifts in response to environmental changes. But Sardans is an ecologist, he's not a chemical ecologist. I am a chemical ecologist, so I gave it a little bit different description. For me, it's chemical ecology plus metabolomics, not ecology plus metabolomics. And you will wonder, yeah, but this is not the same. I don't think it's the same. For chemical ecology, is the study integrating chemistry and biology to examine the chemical interactions among organisms. So we not only study the ecology as an interaction, but we want to go into the chemistry of those interactions, so the signals that are involved in those interactions. So if we mix this with this definition from Sardans, I would say that aims eco-metabolomics aims to study the metabolome, metabolites, and shifts involving ecological interactions among organisms and their environment. That would be my definition for eco-metabolomics. But what is the definition that for Carlos, for the chemical? So I don't want to use definitions of other persons. This is my personal definition. For me, it's just a group of tools. We have a group of tools. We have analytical tools, bioinformatics tools, statistics tools. We have a lot of tools. It's a group of tools that will help us to identify and quantify small molecules, or we call it metabolites, in a given cell, tissue, organism, on an extract in a defined time. This is very important, and we will see today in this introduction, under specific interactions or environmental conditions. So you see that I play a little bit more with a more robust definition of what I think we do in chemical ecology and how we use metabolomics for chemical ecology. Okay? Good. This is what most of the people, when think about chemical ecology, they think about plant scientists. So let's try to use this diagram to think about the interactions that we can face in chemical ecology. So if we think about plant-insect interaction, we have the plants that are attacked by herbivores, that can be sucking herbivores or chewing herbivores, and then the plant has to defend and has a global defense, but also the insect has a global defense against the chemicals of the plant. So we already have a direct interaction here. We can have indirect interactions with some signals are coming and calling predators that will eat those herbivores. But we can also have communication with other plants, with specific plants that will prime the defenses in the neighbor plants. And in these neighbor plants, so these plants will be a little bit better defended against those predators. But we also have below ground defenses in the root. This is also something that I have the impression that when I was starting to study chemical ecology, I always forgot a little bit the below ground situation. And luckily now there's a lot of people trying to face this section of the entire system of plant insect interactions. We can also have pathogens not only below ground but also above ground, and there's a communication. So we have multi-trophic interactions there, and this is something that I'm trying to do in the future, a little bit more authorization of multi-trophic interactions. We also have communication with other type of plants, parasitic plants, competitive plants. We also call pollinators. So as you can see, we have a lot of interactions, but one of them, if we don't have plants, this is mostly what we have. We have an organism that can be affected by abiotic factors or biotic factors. It can be affected by a global change because we have all these different possibilities, atmospheric changes, eutrophication, climate change, pollution, and this will change all or affect all these abiotic factors and will affect directly the organisms. So we have a lot of things happening and going on in our environment that could affect the different organisms. Then we have biodiversity loss or invasive species that also could come and affect the organism and the entire metabolism of the organism. And as we discussed before, we have all these biotic interactions with other organisms, pathogens, below ground, above ground. Okay, so this is what we are trying to face. All these connections, all these communications, all these interactions, how are they affected chemically? Okay. This is what I thought, when I think about 12 years ago, when I thought about metabolomics, this is what I thought. I thought we have a bunch of, it's like looking for a needle in a haystack when we are trying to search the molecule that is responsible for this phenomenon, isn't it? And this is how I felt, and probably some of you must feel right now like that. I say, well, what the hell is that, how I do it? But then when I started studying a little bit all the tools, by that time was only XCMS online available. The NSET mine didn't exist. I think NS Dial was there by that time, probably at the same time. Or not Galaxy, how's the name of the French one? There's a French tool that they. W4N. Sorry? W4N. Yeah, exactly, that one. This is how I felt. I felt myself, okay, I have a big problem, I have a lot of metabolites, but now I have bigger problems because there's a lot of software there that I need to learn how to use. And I felt like stuck with my head in a problem, in a big problem. And luckily for that time, there were some workshops in the XCMS and XCMS online and how to do all, what was metabolomics. And somebody teach me what was metabolomics. So somebody teach me the tools, how to use them and how to find this needle in the haystack. Okay? And this is what we are trying to achieve this week with all of you. Just for you to show you some numbers, if we look for metabolomics in PubMed, I'm sorry, we only find 2 papers. This was 2000, it was 2020, if I'm not wrong. But if we search the individual transplants and metabolomics, we have the highest number of papers there. If we have ecology and metabolomics lower, and so on. So most of the plant papers are plant organismal interactions or plant environmental interactions. And most of the tools, analytical tools in those papers, most of them are LCMS and BCMS in comparison with the other tools. So now you start seeing what is the tendency of eco-metabolomics out there, but it's changing dramatically. So eco-metabolomics has evolved so much that now, look at here, look at 120 people registering this one. So there's a huge number of people trying to use this tool now. When I attend these workshops, we were 10. Okay, so just for you, 12 years ago to now, it's the huge, there's 10 times more people. Good. We are looking for metabolites. This is what we are searching this week. And this is what you are searching in your projects for different metabolites. What we want to achieve is look for organic compounds, look for substrates. Those organic compounds can be substrates, intermediates, or products of metabolic interactions, can be products of foreign substances that get they are intaken or taken by the organisms and they transform it, for example. They are normally below this. If we are a little bit bigger than this, then we are talking about proteins, and then we leave that field to the proteomics. We don't want to touch, and we don't want to step on their field. So we like to think about small molecules, but sometimes we find little peptides, and they are still below 1,500. Okay. What are the main group of metabolites? And this is a common definition or discrimination of metabolites, we call them primary metabolites. They are essential for the basic cellular functions involved in central metabolic pathways. They are normally amino acids, sugars, nucleotides, or organic acids. And we know that, isn't it? But we also have secondary metabolites. They are not directly involved in the fundamental metabolic pathways. They could be they have specialized functions, and they are mainly alkaloids and so on and so on. And this, if we think about plants or photosynthetic organisms, they are mainly divided here. The secondary are the end of these pathways, and the primary metabolisms are somewhere up here at the beginning of the pathways. But actually, this is not so true for a chemical ecologist, because what is a primary metabolism in one organism cannot be necessarily continue being a primary metabolism in another organism. It can become another type of roles. And this is just an example that Matthias Erb put in a paper, and I think it's very nice. For example, glucosinolates. They define 3 different roles of metabolites: primary, secondary, or regulation. And the glucosinolates, if we work in Arabidopsis, they can be the primary metabolism because we can store sulfur or store energy there. They also work in regulation, in root growth. But if we go to another plant, they can also work as a regulator in maize, for example. So depending on the organism, depending on the interaction, the metabolite not necessarily has to be primary or secondary metabolism. It could be regulation metabolism, it could be communication metabolism. And if we look about the benzocosinoids, it's the same story. We can have primary, they can work as a primary metabolism in maize or wheat, but also they can work in Arabidopsis in these regulation processes. So it's not necessarily the same role for the same metabolite among the different species or the different plants or the different organisms. Could have different roles. But also, if we think about interactions, this metabolite that is in the plant has a role as a resistance, then the herbivore uptake this metabolite and maybe make him resistant to predators, but we don't know what is the role of this metabolite in the predator. Maybe it has a role. No, so far I haven't seen any paper asking or trying to solve this question. What is the role of that metabolite in the predator? Maybe it's energy storage, maybe it's not, maybe it's also for his own defense against birds, for example, if it's a wasp. We don't know. So as you can see, we can change and jump from roles of the secondary metabolites or primary metabolism to regulation, depending on where it's the metabolite. Okay, so let's try not to be so square and think about primary and secondary. Let's just think about roles of the metabolite. Good. What is the metabolome? What do you think is the metabolome? You're too quiet. I hope that by Friday we are shouting like hell. But you're too quiet today. You're making sleep all the speakers. So you have to be more active, guys. What do you think is the metabolome? What would you say? The metabolome is
all the metabolites present in your sample.
All the metabolites present in your sample. That could be a good definition. So for me, it's a metro map. So the subway map with all these different pathways, all these different routes, and then we have all these stations here that are very important. And then as many people, tourists in cities like Cologne, when you go downtown, they are with the map and they are lost. And we are actually a lot of times in exactly the same. We are lost with the map of the pathways, trying to figure out where to go and where my metabolite went or in what was transformed, or which is my metabolite. So, yes, metabolome is the sum up of all these metabolites, but why we in chemical ecology focus on the metabolome and not the genome, transcriptome, or proteome? And the main reason, and see, if we think about the connection with the environment, the metabolome is the last step, and it's actually the piece of the puzzle that is having the connection with the environment. These are those molecules responsible for the interactions, molecules responsible for different functions, defense, repellence, attraction. It's not the gene, it's not the transcript, it's not the protein, it's the metabolite. And this is what we think is very interesting, to think about the more physiological part of the entire chain of production of these molecules, and we work with the metabolite. Okay? We talked about metabolomics, and then everybody says, targeted metabolomics, metabolomic profiling, metabolomic fingerprinting. So we know that targeted is when we have a goal, and this is actually a very nice project. Some people who work in metabolites says targeted metabolomics is not metabolomics. And I think maybe you have heard that. Metabolomics is if you don't know targeted. Targeted is just a simple analysis of metabolites. This is not metabolomics. I don't want to go into this discussion. I would just include it today as targeted is part of metabolomics, and profiling and fingerprinting is part of metabolomics. For me, this is targeted. When you look for Wally, and you know who are you looking for, and it's a little bit more simple. Let's say it's not as simple as this. But it's at least you know what are you looking for, and most probably you already have found in previous papers how to do it and how to achieve to search for that. Sometimes it's not so easy. Sometimes you try to reproduce what is in a paper and actually never works. So that's actually what almost always happens. This is for me profiling. So you want to know a little bit of group of metabolites. It's a little, I would call it this a little bit medium targeted, non-targeted, because I want to profile the lipids, for example. And this is another dark area that people from from lipidomics says that they are not metabolomics because they do lipidomics. For me, for Carlos, they are part of metabolomics. They just analyze a different type of molecules that are called lipids. But, and they are big, yes, but we see lipids in metabolomics all the time. So, but some lipidomics, hardcore lipidomics people say, no, we are not lipidomics, we are lipidomics. Okay, so yeah, you try to find Wally probably in this profiling, or maybe you know something about what you're looking for, maybe you don't, and maybe you find Wally there in the middle of your profiling. But what I personally like is this, is the non-targeted metabolomics. This is what I think this is what I have been doing my entire career. People come to me and say, hey, Carlos, we have this idea. Do you think we can do something? We can find something there. And this is the challenges that I like, that I really enjoy trying to find. They are very risky. They are long time. You require a long time to find something. Probably you're very lucky and you find something. Probably you're very unlucky and you don't find something. But for me, it's very exciting. And many of these tools that these guys have evolved and has developed as actually to help us to find those models. So I found Wally, and luckily for me in this screen, I found Wally, but isn't that always the case? Sometimes I have to search many times for Wally. What are the different metabolomic approaches? And then here I will tell you what I think is the metabolomic approaches, because there's different approaches, so different ways to approach the approaches of metabolomics. I'm not here. I would consider, as a chemical ecologist, I would divide metabolomics into different broad ranges in endometabolomics and exometabolomics. That's for me the two bigger approaches of metabolomics. Okay, endo, people call it also fingerprinting, and exo, people call it footprinting because it's what the species or the organisms are releasing and are laying in as a footprint on the path, wherever they are around. We can have both of them targeted or non-targeted. You know that. And both of them can be done by mass spectrometry or by NMR. This week, we will focus in mass spectrometry. Isn't it, guys? Fully concentrated in mass spectrometry. I'm not a NMR metabolomics person, and I think we have designed a course specifically for those guys, for LCMS, for MS metabolomics. Okay? Good. This is, if we have the interaction between a plant and a herbivore, we can analyze the endometabolome of the plant. This endometabolome, or we can analyze the exometabolome of the plant. In this case, will be the volatiles released in the environment when the plant is attacked. We can have targeted metabolomics, so we can go and search one specific endometabolite or exometabolite. Or we can have non-targeted, also trying to check as much as we can, or try to make obtain or extract as much as we can from these. Metabolomics is not only for chemists, it's not only for ecologists, it's not only for biologists. We have, and we are lucky that we have a big group of scientists. It's very interdisciplinary. We have biology, analytical chemistry, bioinformatics, statistics, and this week you will have all these persons giving you talks. So this is pretty cool because we will have different ways of thinking also, how we see metabolomics from our own areas and how we think we integrate it in the other areas. So you will see that our way of thinking, although we work with the same goal, sometimes the way that we see the things from our perspectives, from our careers, it's a little bit different, but we try always to work interdisciplinary and to mix them. That was how was done before. A long time ago, it was done by bioassays. So people used to do a bioassay, and they found that an extract of a plant was active and was killing, for example, a weed. And then people do fractionation of this extract of this plant that was active. And then they have to do bio. So here I put this time because the bioassay requires time, especially for those of you who work with plants or with big animals. You will have a lot of problems because there's some plants that take 4 months to grow with this dry stage. And then if you set up the entire experiment, maybe you have 6 months for just one single experiment. So this is time here. Bioassay means time. And then you do the extraction or fractionation, and then you set up another bioassay to test the fractions. And maybe you find one that is active, one fraction that is active, but then you try to fractionate again to find where in this fraction is the metabolite, and then you fractionate it again. So more time, more time, more time. That was what we used to call bioassay guided fractionation. That was the classical method. It's still used. We still use it. We integrate it now with metabolomics. It's a very useful method, but I think with metabolomics we can speed up things a little bit more. Why? Because now we have extracts. We test. We found one extract that is active, one that is not active. Then we take the metabolic profiles. Then we use all these beautiful tools that you will use this week, and try to figure out what is present in the active that is not present in the active, or what is increased in the higher in the active that is not in the inactive extract. And then you can fractionate here and try to purify that compound and do a balance. Less time, isn't it? Less bioassays. And it's even better when you have a pool of different extracts with different range of activity, because that will help you to compare all different range of activity, and then you can confirm that your metabolite over the different range of activity is increasing or decreasing. It depends on you. It depends on your question, but then it's easier because we will really see that this metabolite is responsible for that. You will try to purify the metabolite or try to buy the metabolite and then do your bioassay. So you see, we jump from 3 big bioassays to probably only 2 bioassays. So things that used to take 5 years, it can take now 2 years, probably to find something very nice. Okay? Good. This is the workflow that we know from metabolomics, and this is what I'm gonna go. I have still 20 minutes. We have different sources of organisms that we want to take samples. And so far, I think I checked most of your profiles, and I have a pool from plants, microorganisms, environment, food, humans. I actually see some human-related people working on human metabolomics or human science, and I think this is cool. We will not talk too much about human during this workshop because it's not my experience. I don't work with humans, and I think we don't work with humans, So we are more into the environmental plants, organisms, soil metabolomics. But there's eco-metabolomics also in humans with the microbial communities in the skin, in the stomach, all this interaction that these microbia have in the stomach. But we are not experts on that, so we will not touch humans during this week. But most of the tools and most of this workflow that we will show you in this week is completely, you can also apply it for human research. Okay? Then we will extract our samples, go to LC or PDC, beautiful instrument, expensive instruments, collect our data, separate our ions, and then we will have our chromatogram and our masses, and then we reach here, and then we will call Stefan and say, hey, Stefan, I need to want to use MZ-Mine to get some data from here because I don't know what to do with it. And Stefan tomorrow morning will give us a beautiful hands-on session how to get data from here, because this means a lot. There's a lot of information there, and we can collect that information. Okay. This is what I'm gonna use for this introduction. What is the workflow? What I think is the workflow. First of all, biological question. I think Josh will talk a little bit about biological question. I just want to say, I hate this. When people come to me and tell me, Carlos, can we do metabolomics with that? And I think it's not only me, the one who is suffering that. I think many people who work with metabolomics have the same problem, that there's people, because they think they go to the field and they collect whatever they want, they can come to us and tell us, Carlos, can you do metabolomics with this? No. And today I hope, after the talk of Josh about design, after the talk of all of them, that metabolomics has to be designed from the right moment that you think about doing metabolomics, before you go to the field. It has to be planned. It needs to have a right question about metabolomics, okay? So keep calm and please formulate your research question very carefully, because if not, you cannot do metabolomics. Okay, so you need to know what do you want to know, and maybe sit down with one of the guys that knows about metabolomics in your institute and ask him, hey, can I really answer? Can metabolomics help me to provide some information there? Because maybe not. Maybe we are not able to provide you with something. Maybe our techniques, our tools are limited for the questions that you have. Okay? Can we validate our findings? And this is very important. Although I think maybe Daniel, that is online right now, will disagree with me, but there's a lot of explorative projects going around. So I have participated in explorative projects without any thought about validation. We just do metabolomics, take a bunch of environmental samples, following the replication and careful randomization, like Josh will teach you later, but we don't know what is there, because nobody has done anything before with those samples, and nobody has explored there before. So I think Daniel also has done things in ocean, with ocean chemical ecology, and it is basically the same idea. So some people just go there, take samples. We think we can we want to see if there's differences between different spots, different depths, different type of samples. These are more explorative, and they are really, really risky projects, but sometimes they come up with really, really good, nice, awesome results. Remember, it's better one answer than a clear question than a guess at three. So this is something that I try to tell all the people who comes to me and tell me I want to do metabolomics. So it's better to have a real question than try to answer that question. Then experimental design, and then we will have Joss just right after me in 20 minutes. Joss, I will introduce. Joss, will you introduce yourself? Yes. Okay, but I just want to tell you, I met Joss 3 years ago in another workshop in Wageningen, and I love the talk of Joss. And yeah, so
we were talking about biological questions and environmental samples. So if you get an environment, you don't know so much because I work on glaciers. So we work on the algae that grows on the glaciers. So we don't know much about the algae on the glaciers and their metabolics and their roles. So how could you suggest, like, if you want a hypothesis or a biological question, how to design? Like, if you start paper, there is no literature. Like, nobody works on that. And now we are trying to figure out, we are doing some stuff. So how do you suggest, like, from metabolomics point of view, how to design a hypothesis or biological questions? Because we just take samples, we quench the samples on the field, we bring it back, dry, and then do contaminate metabolomics, and then we are working to see different metabolites, and if you can link with anything from the literature. But there are not so database and anything for any literature. So how would you suggest?
we are the same team. We are the same team. We work with Daphnia. There's not much going on Daphnia with metabolomics, mostly for ecotoxicology, and we are trying to do metabolomics on Daphnia for a completely different purpose, and we are struggling as well. And we also, when we try to sell our projects to the EFG, this is the main criticism of our projects, that they don't have a hypothesis. So remember that non-targeted metabolomics is a hypothesis-generating approach. So the idea of doing non-targeted metabolomics is that you will generate a hypothesis from the results of your approach. Okay? You can sell it like that, but I think Josh will have a better answer for these kind of questions than me during his talk. So what to do there, how you can try to generate open, very broad questions regarding that. But Josh is the expert in experimental design.
Talk at a coffee break, maybe.
You can also get a coffee break.
Down.
So I personally, so solution that we got is, of course, you always try to please the reviewers of this DFG, and then you try to sell it. Okay, what we will do is try to validate the findings of somebody else. That will be a way.
Yeah, it's the same problem. Like, reviewers are like, how can we say there is no literature? And then like, yeah, they publish 2 papers in the whole project.
It's very difficult. It's very, very tricky. It's very difficult to sell the idea as well.
Yeah.
But it has to be done. Somebody has to do it the first time, you know? And I think the best way to do it is with non-targeted metabolomics, to try to set up a path, to find at least a chemical class where to go, or to find a metabolic pathway that we can search for and we can work with. It's tricky, but I personally would suggest try to validate the findings of somebody else, or somebody has made something very, very similar. Try to use this to validate the hypothesis that we're generating here. That's the only way that we found to answer the reviewers that were giving us those criticisms about our projects. But we are the same team, exactly the same team. We have those issues every time that we submit a project. Okay. Good. Josh comes from the University of Wageningen, and I will not say nothing about him because he wants to introduce himself. But luckily for you, you have one of the experts on experimental designs, biostatistics, chemometrics. You have a long list of expertise, Jim. So all the questions that you have, you can also do the breaks, go with Josh and ask him if you have a project, you have a problem with the statistics. Josh actually has helped me a lot with the statistics when I ask him some things. Josh will go for the experimental design. Then we will have sampling and sample preparation. That's me. I don't want to introduce myself. We will know each other during the coffee breaks. Then, as you may know, we have that acquisition, and Allegra will give us today in the late afternoon a beautiful talk about that acquisition. She has been working with LCMS and GCMS broadly, so she has a lot of experience about that acquisition. And she works with this chemical biology of metals and metal binding molecules. And we are very interested to hear a little bit of that because it's a new technique for metabolomics, how to use metabolomics into this field. So I'm really looking forward to hear from her all what she has to tell us. Then we have Stefan, who is sitting here. Stefan is one of the main developers of Edinset Mine. So there's actually two, isn't it? Stefan and Robin. Unfortunately, Robin was planned to come today, but right now, at the same time, is happening the mass spectrometry conference in Munich, of the German Society of Mass Spectrometry. And these guys, luckily for us, decide to split. Stefan is in Munich. I'm sorry, Stefan is here. Robin is in Munich attending the mass spectrometry group. So thank you very much, Stefan, for being here and for taking your time and missing all the mass spectrometry information. Stefan works in the University of Münster, and he's on. I can tell you, Doctor.
Not yet.
Not yet. Okay. But actually, Stefan just submitted his thesis, PhD thesis, so soon we will have a new doctor from the field. Then we will have Giovanni. Giovanni works at the University of Tübingen. He's a postdoc researcher there. And he will talk about, he will start tomorrow, no, the day, yeah, tomorrow afternoon, he will start the part of data analysis. He has been working with GMPs and feature-based molecular networking. I always mixed those ones. He has been working in the lab of Professor Daniel Petras. Daniel Petras will be here tomorrow if the strike allows him to arrive at some point. They belong to the lab of functional metabolomics in the University of Tübingen. But Professor Daniel Petras now is in UC Riverside, and actually I have to talk with him because that was new information for me when I was designing all this workshop that he was there. But yeah, Giovanni will introduce us to this. Giovanni has a lot of experience. He has been helping us also here with some of our projects. We have cooperation with him and with ACER. And then we will have Professor Min from UC Riverside. He just decided and do GMPS2 for those who know GMP. Now yes, GMPS2 exists, and we will have the pleasure to listen to the person who developed GMPS2 on Thursday late afternoon in a remote talk. He will tell us everything about what is the advantage of GMPS2 compared to DMPS. So I'm actually also looking forward because I don't have any idea what is. I didn't have time yet to play with GMPS2, and that's why you have these tokens. I think you got my email in the list that I share with you. I also share the list of all of you with the names and institutions and the emails in case that you want to keep in touch after the workshop. And in front of you have the tokens. Please use this individual token that was assigned to each of you to have access to the GMPS tool. Okay. Something else about the tokens, Giovanni?
No, yeah, do it before tomorrow, because tomorrow we will like play this with the VHS too, so we'll be like an interacting thing. So please do it before tomorrow.
Yeah, it should be done before.
The token you can like create like your account, because it's not public yet VHS2, so you just through this token you can create your account, and then we can play together. We can try to put together our molecular method.
So this is one of the advantages of this workshop. GMP2 is still not available for the public. We got a special access to GMP2, and we got specific tokens to each of you to get access to GMP2. So once GMP2 is open for everybody, you will have already previous knowledge how to play with GMPs. Okay. And then we have ACER sitting here in the front and helping us also to record all these talks during the workshop. ACER is the awesome author of this beautiful paper, and she has built a beautiful platform for the hitchhiker guide to the statistical analysis of feature-based molecular networks for non-target metabolomics data. When that paper came out, and I had a look of that, and I think, finally somebody did it. You know, finally somebody thought about people like me, that I'm not a developer of XMine or a developer of GMPS. Somebody took me in a paper all with my hand and through all the steps with this challenging process of data. So thank you, Abser, for doing that. And she will show us how to do it here in person. This will be Friday, all Friday. She's a PhD student actually at the University of Tübingen in the Functional Metabolomics Lab. And then we have Professor Daniel Petros, who also will take all this data, annotation data, and will put it together and will show us in Cytoscape how to put all this data together and how to get more from this annotation and GNPS networks and everything. Daniel will arrive tomorrow, so hopefully the trains will take him to Cologne. Marcus was one of the main developers of Sirius during his PhD thesis, and now he is one of the CEO of Bright Giant. Bright Giant is just a company that they built, it's a spin-off from the university, and now they are providing Sirius adaptations, I would call it, for industry. And so now the series that we use is the one that the public one, but they produce like specific tools for industry right now. He will be with us tomorrow as well. He will come and share, because on Thursday morning, he will teach us how to use Sirius for annotation and how to get the best from his tool. Okay, so we will have all the annotation part. And then we will have the biological interpretation. And here I forgot actually that I want to take a picture of all of you before and put it here, because this is what we will do. You will do the biological interpretation. So we will be sitting here. We hope that Daniel, Giovanni, Abser, Stefan, and Josh will help us a little bit to understand. Let's try to get in this data example that we provide, so in this data that you already hope you downloaded, then you can find a little bit what we have. And just for your knowledge a little bit, I use normally to go to each one of these 7 points. These are the 7 points that I thought, and I think, how metabolomics can help in chemical ecology. And I have in my slides, I will put my slides in the driver, and then you can go through each one of the examples for each one of these. I don't have any more time. I have only 5 minutes left. So just for your information, we can work in trying to find chemical signaling, and I think this is the most common one. What is the chemical signal that is involved in this interaction, and what is the attracting, repelling, killing, all this type of signaling. We can also identify bioactive compounds, not only that signal, but also have some kind of activity, deterrent, toxic, suppressors, apoptotic molecules. So there's a bunch of bioactive molecules here. We can also do eco-metabolomic fingerprints. This is more for the non-target group. Our team, the non-targeted guys that we say, okay, let's do metabolomic fingerprinting and let's see what is there. So this is a little bit more broad biodiversity and try to search what is in the environment. We can also unveil hidden interaction. This is for the people who work with symbiosis, microbial communities inside an organism. It could be symbiote, it could be non-symbiote, but there's a lot of hidden interactions that there was a long time that nobody thought about these interactions. So it can be used also. We can track responses to environmental changes. There's a lot about this now, especially with the global warming and the climate change. So there's many people trying to check what happens when the global warming, with the heat, with the ocean acidification, withdrawal, with salinity, what happened with organisms when all these changes, environmental changes are occurring, or try to have a perspective, what is going to happen if this happens in the future. Then we can also unravel trophic relationships, and this is something actually that I'm looking forward to starting to study. What happened if I put this here, and then what happened with this host plant, for example, and what kind of results are going on with the predators or with other plants or with other organisms below that. So this is, I would say, probably one of the futures of chemical ecology, to start integrating multi-trophic levels in one research. And finally, conservation and biodiversity, that also makes part of this non-targeted profiling. Okay? Do you have any questions? Because we finished. Yes.
Sorry, I really don't have a question, but the kind of which, first of all, thank you for organizing this kind of workshop for us. I have seen that it's really loaded. I am really looking forward to all the things that are attached to it. Because unfortunately, I have a bit of challenge. I won't be to the end of the day because I actually have to pick my kids in the. So is it possible? Because I really don't want to miss any part of it. Maybe like a recap or.
Yeah, we are recording every talk. Every talk will be recorded, and then I will send you an email that you will have a link that you can get access. Or because in the drive, I don't think we can put all those talks. Maybe it's too heavy for the drive, I'm not sure, but we will figure out how to communicate with you where you will have access to these talks. Okay. So all the talks will be recorded. We will do our best to record them with sound and image. So we will do our best. Okay, but everything will be. You're not the only one. The people who couldn't participate or they couldn't come because of the strike of the Deutsche Bahn, they also were asking in the last days exactly the same question. Okay, and I'm sorry if I didn't answer emails, but I got so many emails between the last 3 days that I couldn't reply to all of them. I'm sorry, I was super busy organizing here. Okay, good. Any more questions regarding chemical ecology? Nope. Good. Thank you very much for your talk, for your attention. I'm sorry, I'm a little bit ill, so my brain is not working properly. I will invite then Joss to the stage, and Joss will tell us about sample experimental design, and the stage is also yours. Thank you very much.